Hello everyone, welcome to ITLS Academy. I am Ronak Fadma and today we will discuss about the anaerobic bacteriology. Now let's start with the basic definition of anaerobic bacteriology. Anaerobic bacteriology is defined as a branch of microbiology that specifically deals with anaerobic bacteria. All right. Now, what is anaerobic? Anaerobic is defined as organisms or processes that can function and grow without the presence of molecular oxygen. Without the presence of molecular oxygen means the oxygen is absent completely or poorly present. Alright, now how anaerobes are different from aerobic bacteria. So let's learn the differences. Unlike aerobic bacteria, anaerobes generate energy by the process called fermentation, and aerobic bacteria uses aerobic respiration method. All right, anaerobes lack respiratory enzymes for oxygen. This means they cannot use oxygen as terminal hydrogen acceptor. All right, most of the anaerobes survive in the range of 3 to 5% of the oxygen level. However, some anaerobes are very sensitive to oxygen level and they can only survive as low as 0.5% of the oxygen level. Alright. Some anaerobes have tendency to grow but at very poor level in the presence of oxygen, and they are also called as aerotolerance anaerobes. Alright. Now, many are the part of normal flora that are supported by the facultative anaerobes. Alright. Normal flora normal flora is defined as the community of microbes, that means microorganisms that live or live on or live inside the human body or the animals without causing infection or diseases. All right. They are also called as microbiota or microflora. Okay. And let's learn some of the basic points of anaerobes, such as uh, anaerobes produce organic acids, alcohols, and gases as byproducts. All right. Now, one of the basic question arises: where are anaerobes found? Okay. So there are three major locations they are found in. First of all, human body. Where in human body? Gastrointestinal tract, oral cavity, vagina, or skin folds. Okay. Now the second location is soils or sediments where oxygen level is very poor, such as deep mud. And the third location is industrial environments, such as where there is occurring waste treatment or there is the production of biogas. All right. Now comes the next slide. As we have discussed, anaerobic bacteria, just a second. Yeah, anaerobic bacteria form an important part of our normal microbiota. Okay, they are present on most of our body parts, body surfaces, and mucous membranes. They are found in large numbers throughout the gastrointestinal tract, as we have discussed. Mostly large number from mouth to colon, but in comparison to mouth to colon, they are lesser in stomach and esophagus. Now, also they are very abundant. That means they are also present in large numbers in the female genitourinary tract. Now, what are the what is the relationship between anaerobe and humans? First of all, um, humans provide environment, a secure environment, a stable environment for anaerobes, and food, water So in return, anaerobes helps us with digestion and prevents harmful microbes that are also called as pathogens uh, for causing uh, infections by attaching. Okay, and also they are the part of our natural immunity. In addition to this, they also supply essential nutrients like vitamin K and cofactors that cannot be naturally produced in human body. Vitamin K is also works as cofactor for enzyme called gamma glutamyl carboxylase. Okay, the gamma glutamyl carboxylase. And how it helps? It helps human body in blood clotting, in bone metabolism, and also in the activation of calcium binding protein. Okay, now in the next slide, we will discuss about the classification of anaerobic bacteria as per the tolerance of oxygen. Okay, anaerobic bacteria are basically categorized in three forms: obligate anaerobes, aerotolerant anaerobes, and facultative anaerobes. Now, what are obligate anaerobes? Obligate anaerobes are the anaerobes that can be killed by oxygen. Okay, that means they need completely oxygen-free environment to survive and cannot grow in the presence of oxygen. Okay, now, for example, Clostridium, bacteroids, and Actinomyces. Okay, now, what are aerotolerant anaerobes? Aerotolerant anaerobes are defined as the anaerobes that can tolerate oxygen to a limited time period but cannot use it or depend on it. Okay, for example, Lactobacillus species or and Streptococcus species. Okay, next comes facultative anaerobes. Facultative anaerobes are defined as the anaerobes that have potential to grow either in the presence of oxygen or absence of oxygen. For example, E. coli, Staphylococcus species, and Salmonella species. And when facultative anaerobes survive in the presence of oxygen, they use aerobic respiration method. And when they survive without the oxygen level, they uses fermentation method. Okay. Now, anaerobic bacteria and their associated infections. Normally, um, anaerobes are harmless, uh, but they can be pathogenic when these three conditions are applied. Okay. First, when they are overly grown in their usual sites. That means the normal places they generally occurs. Okay. The next, the next condition is if anaerobes are introduced in a new sterile sites, that means the sites, the location, they are generally not present. Okay, and this is called endogenous infections. Okay, and the third condition is when anaerobes from outside enters the body via wound infections, uh, when trauma occurs or surgical methods occurs or through accidents. Okay, so the method called exogenous infections. Okay, now anaerobic infections include wound infections uh, that occurs generally after trauma, surgery, and abscesses in liver. Abscesses, we say swelling with pus in liver, brain, lung, appendix, and peritoneum. Peritoneum, abdominal lining. Okay. Chronic sinusitis, otitis media, and dental infections. Now, these three are interlinked. Okay, these three are interconnected conditions where infections are spread between nasal cavity and middle ear and oral cavity due to sheer anatomical pathogenesis and proximity. Okay, And now comes severe systematic diseases that involves endocarditis. Endocarditis, infection in heart valve. Bacteremia. What is bacteremia? Bacteremia is defined as the presence of viable bacteria in the bloodstream. Okay. And myonecrosis and gas gangrene. Now, myonecrosis and gas gangrene are defined as the severe muscle and tissue infections. Okay. Now, let's learn about the incidents related to anaerobic infections. Okay. Before prophylactic antibiotics, that means prophylactic antibiotics are the antibiotics that are generally given before the surgical methods to, uh, to as a precautionary method to prevent diseases or infections. Okay, not to treat them. Okay, so before in early phases, uh, before the use of prophylactic antibiotics, anaerobic bacteremia incidence, the disease incidence reached to 20%. Okay, but today when prophylactic antibiotics are introduced before and are given before surgical methods, we have observed that there's a decline in aerobic bacteremia incidence from 0.5% to 9% with an average of around 4%. But depending on the patient's age, site of the infection, that means location of the infection, and the population demographics. Okay. Now, anaerobes rarely cause UTI infection. Okay. Now, also, they rarely cause meningitis. Meningitis, what is meningitis? Meningitis is defined as inflammation of fluid and membrane around bone and spinal cord. Okay. And also, anaerobes rarely cause diarrhea, except for the Clostridium difficile associated diarrhea. Okay. Now, the currently, reports have been seen the increased rate of such diseases. You can say the rising of Clostridium difficile associated diarrhea cases are already increased, and the resurgence of Lemierre's syndrome. Now, what is Lemierre's syndrome? Lemierre, sorry, Lemierre's syndrome. Lemierre's syndrome is defined as a rare but a serious infection of a throat that can lead to septicemia. Okay. Now, uh, also there are several reports uh, of the discovery of newly recognized anaerobes that are linked with several types of infections. Okay. Now comes the slide of optimal recovery of anaerobic bacteria. Okay. So, what is the major step of recovery of anaerobic bacteria? That is, first, just a second. Yeah. Proper sample handling. Proper sample handling is very crucial step to recover anaerobes. Okay. So clinical microbiologists in the microbiology labs should establish a clear guidelines for sample collection and train clinicians and uh, and the workers who collects the sample in the hospitals. Generally, preferred sample are fluids, aspirates, and tissues, but we will discuss in detail about the acceptable and non-acceptable samples in detail in our next chapter. Okay, you can say next part. Now comes to the next point. Swabs are very discouraged because of the following reasons. They have limited surface areas. Okay, they have increased risk of contamination by normal flora. That means there are present of multiple microorganisms, and therefore there are chances of contamination. Okay, and the specific bacteria cannot be examined. And the third point is poor anaerobic transport atmosphere. It is important to provide specific transport atmospheric condition for sample handling for optimal recovery. Okay. Now, if swabs are used in certain conditions, so it must be transported in specific anaerobic transport devices. Okay. And if the processing delays by 2 hours or more than 2 hours, it should be kept in the room temperature and should not be refrigerated. Okay. What are the media requirements? Media requirements are culture media should be fresh and with vitamin K and hemin. Okay, in normal cases. But in some cases, we need enriched media with formate or fumarate. Okay, now generally solid uh, media are used, but broth media can also be used, but their study is very contradictory. That's why in general solid media are considered, but broth media can be used in special cases because if we use it in general terms, they have increased risk of contamination, and we should be very cautious about the contamination risks. Okay. Now let's learn about the direct examination of anaerobic culture. Okay. First of all, uh, know that the direct examination method is very crucial step for anaerobic culture. Okay, and just a second now. Okay, gram stain results must correlate, or you can say must match with isolated organisms. For example, let's learn with example. In CSF shunt specimen, in specimen of cerebrospinal fluid shunt, if we find Propionibacterium acnes or anaerobic gram-positive coccus, it may still consider to be insignificant in case WBCs or bacteria are not visible. Okay, but on the other hand, if gram-positive non-spore-forming rods are seen and polymorphonuclear leukocytes, that also we can say polymorphs, but not in the aerobic culture, then the uh, Propionibacterium acnes or bacteria uh, uh, can be considered as relevant. Okay, now matlab, their findings can be considered as relevant. Okay, now the in the liver abscess, gram stain shows polymorphs plus intracellular gram-negative bacilli plus extracellular gram-negative coccobacilli. If Klebsiella pneumonia and Bacteroides flagella are cultured, both are considered as pathogenic. Okay. Now, sputum is not suitable for anaerobic culture uh, because of its contamination with oral flora. Okay. There are multiple microorganisms present in the oral flora that is mixed with sputum. Okay, so that's why sputum is generally not considered. Okay, but gram stain of sputum showing polymorphs plus gram positive and gram negative anaerobic like bacteria, it suggests that uh, there is anaerobic pulmonary infection and treatment is guided without any specific ID. Okay, but in sterile sites, that means uh, through blood or abscess, complete identification of anaerobe is necessary. That means there are two things. If uh, there are like special if the anaerobes uh, are identified from the sterile sites such as blood or abscesses, complete identification of the anaerobe is mandatory. And other than the sterile sites, if we get uh, anaerobes, then the specific identification ID is not mandatory. Okay, and complete identification is not mandatory. They can be categorized as their clinical relevance. Okay. Now we have to take care of some points for the detection of clinically important anaerobes. Okay, first we have to know about the practical steps to confirm the presence of clinically relevant anaerobes. Okay, so for the detection of clinically relevant anaerobes, we have to uh, check that there must be correct collection and transport of anaerobes. Okay, the next point we have to take care of, we need to accept and reject specimen types based on the contamination risk. Okay, such as we cannot accept sputum, and as we have already discussed in our previous slide, and we will discuss more in detail in our next part. Okay, what are the main with names with their details, we will learn about what are the acceptable and non-acceptable specimen types. Okay, now, the next point we have to consider that uh, the planting and incubation of anaerobes must be done properly under the anaerobic conditions. Okay. Now, direct microscopic observation is crucial with gram staining correlation. Okay. And now the next point we have to consider is initial characterization with presumptive methods are confirmatory identification. Okay, and biochemical tests are also good. Okay, now spot tests for rapid detection are preferable, and it provides the identification of anaerobes in less time. Now, there are some advanced methods introduced, such as MALDI-TOF mass spectrometry method and the molecular techniques for rapid, precise, and for rapid and precise identification. Okay, we will learn more in detail about detailed version of the collection, transport, and processing of clinical specimens for anaerobic culture, culture media for anaerobes, and incubation techniques for anaerobic bacteriology specimens in our next parts. Okay, so stay connected to ITLS Academy, and if you have any query regarding this class or any of the class, you can connect with us through any social media platforms mentioned on the slide. Okay, thank you very much. Let's meet in our next class of anaerobic bacteriology.